plan for today is to discuss the next taxonomic group. We'll spend today on the fungi, and then on Monday we'll start on the next group, the animals. So before we start on the fungi, though, I'm going to go ahead and do a quick pseudo-review question. This is something that should be really straightforward and even easy if you have mastered the major plant groups. So the question is, and the answer counts, so you get more credit if you answer correctly. What kind of plant is an oak tree? If this is a tree you're not familiar with, I've got a picture right here on the right. And if you can't see what the leaves look like, there's a close-up of a leaf, in case that helps you. Okay? So we learned about 4 major groups of plants, right? Non-vascular, seedless, vascular, gymnosperms, and angiosperms. Which kind of plant is it? Chat with your neighbor. I'll give you 20 seconds or so. Then I'll check back in with you. Okay, everybody had a chance to click in? I'll show this number right here. Super. Whoa. Okay, I'm hoping for 100% right. I would consider this extremely easy. You've mastered the trees. Oh, no! Well, I guess I was wrong. Yeah. Well, good job, 61 of you. Why did I show you a leaf, by the way? Because are there a lot of gymnosperm trees? Yes. These trees do tend to have needle-like leaves, and so evergreen trees, right? You know, things that look a little bit like this, right? Christmas tree types. Yeah, nope, angiosperm. Good job, most of you. Yeah, and okay, well, fooled me. I thought that was an easy. Okay, you might want to review your plant groups. Okay. So we're going to consider the fungi now. I pulled this graphic from the BBC website. That's a British Broadcasting Corporation, and because this title caught my eye, the largest living thing on Earth is a humongous fungus. Forget blue whales and giant redwood trees. The biggest living organism is over 2 miles across. So you might be thinking, wow, where is this thing? Why have I not noticed this gigantic 2-mile fungus? Where is it? Okay, well, let's try and figure that out. But first, let's go ahead and consider, since we're going into a new taxon now, how that taxon, the fungi, relates to the other taxon we've been learning about. This is figure 32.7 from your textbook. It is a phylogenetic tree, which shows some branches on it. And I think one of the things that students often find very surprising when they start learning about fungi, by the way, notice this fungi is singular, fungus is, fungi is plural, fungus is singular. One thing that surprises students a lot is, I think for most students, if you were told the 3 groups, fungi, plants, animals, you know, what do you think is more closely related to which? I think most of us would say, well, clearly, fungi and plants must be more closely related to each other because after all, they're way more similar, right? And what are some of the features of fungi that are similar to plants? Well, they are not motile, at least not usually, meaning, you know, they're attached, they don't move around. Same thing with plants, right? And so many aspects of their life, their physiology, their life cycles, is associated with that really important characteristic that they are not locomotory, unlike most animals. Not all animals are locomotory as well, but are motile, but many are. Certainly the more familiar ones. So why is it then that in fact, fungi and animals are much more closely related to each other, right? Here's our most recent common ancestor between the animals and the fungi, and then green plants are way over here as a very, very distant relative. The relationships reflect the major differences in how we make our living. How do we generate energy? Where are the sources of our organic molecules? And we had a little bit of an introduction to diversity in metabolic, metabolism in different groups when we were discussing the microbes. But let's consider now some of the important similarities between fungi and animals. And here's a partial list for you right here. This is the same phylogenetic tree. I just shrunk it down to get it out of the way so there's room for some text here. So the most important and significant similarity is, in fact, where we get our organic molecules. So both animals and fungi are heterotrophs. Again, this term should be familiar to you from when we were discussing metabolic strategies in the microbes. If you have forgotten these terms, good time to revisit them so when these terms are used, you know what they mean and you can interpret things correctly. So both fungi and animals are heterotrophs. We need complex organic molecules from other organisms because we are incapable of just simply synthesizing them all ourselves. Now, there is a difference, of course, between how we get our complex organic molecules. Animals, for the most part, not all, but most animals, eat. We consume other organisms in order to get the organic molecules that are locked up in the bodies of other organisms, whether they're plants or other animals. Fungi are completely different. They actually absorb the organic molecules through their body's surface. Completely different from us. Okay. So that makes both of us, though, heterotrophs, even though our way in which we bring organic molecules into our body is a bit different. We both have a complete dependence on organic molecules that were, in fact, are from other organisms. Another similarity is that most fungi and most animals synthesize a very important and extraordinary structural material called chitin. We actually, humans and our close relatives, don't have chitin, so this is not something that humans have. So I'll go ahead and put humans don't. So you're wondering where your chitin is. You don't actually have any. Most animals do. So for example, if you think of insects, part of the outer covering on insects is chitin as one of the individual chemical components. And so most animals, even though we don't, most animals do in fact have the ability to synthesize chitin, and it's part of their bodies. Fungi also, most, although not all, synthesize chitin as well. So that's another similarity. Another similarity is similarity number 3 on this list, and that are similarities between the flagella. In fact, as you're looking, as you're becoming more aware of some of the different fungal groups and fungal lifestyles, you'll see that many fungi, in fact, most fungi don't even have flagella at all, or if there are flagella, they're just in a couple of parts of the overall life cycle. But for those fungi that do have flagella, they're very, very similar in structure and composition to the flagella that, in fact, one sees quite commonly in animals, including humans. And so, so where are flagella in humans? Think, sperm tails, for example, right? That's how sperm swim. They're flagellated. Those are flagella. So that's where flagella occur in humans. So the flagella are very similar in fungi and animals and distinct in many ways from flagella that are in other organisms, such as the protists, for example. Another similarity between fungi and animals is that polysaccharide storage is done with a chemical called glycogen. This is a polysaccharide, and this is completely distinct chemical form from how plants store carbohydrates. The polymer they use is starch. So starch is a completely different chemical compound, so the actual synthetic routes are different for making starch versus glycogen and so on. So very, very different for storage of carbohydrates. So that's another similarity between us and the fungi. There are several major lineages of fungi. They're all quite distinct from each other. They are diagrammed in the phylogenetic tree shown here in figure 32-8 from your book. The major differences between these lineages are ways in which spore-producing structures are really quite distinct and are shared synapomorphies within many of the different lineages. You absolutely do not have to memorize the names and distinctive features of all the different lineages. So do not worry about that. We'll certainly go through some specific examples of interesting fungi to give you an idea for the diversity and significance of fungi, but you do not have to learn all these different groups. So let's consider when the fungi evolved, and we'll do this with a visual backdrop of the usual Phanerozoic eon, figure 28-7 from your textbook, which is in front of you right now. Hopefully at this point, since we have discussed several major milestones in the evolution of life on the planet, you can approximate where various things occurred. Right here is the Cambrian, the time of the Cambrian explosion. Right here is the next period in time, so one period more recent than the Cambrian, which is when the very, very first land plants, perhaps those moss-like non-vascular plants, invaded the terrestrial landscape. But when exactly did the fungi evolve? Well, it's pretty clear that they invaded land either with or very soon after the very, very first plants, meaning the non-vascular plants, the very first inhabitants of the terrestrial landscape that were multicellular at least. But presumably they evolved before then, and so it is thought that fungi evolved sometime prior to this time. But during this earlier time period, the pre-Cambrian, there is very little in the way of a fossil record. So it's there's very little information about the very first fungi, but it is very clear that they were on inhabitants of the terrestrial landscape pretty soon with or shortly thereafter the very first land plants. Okay, so let's go back to the topic of how do fungi make a living. Okay, and again, fungi are not motile, they don't run around, and so if they're going to absorb nutrients from other organisms, then they have to actually be in physical contact with their food source. The organisms from which they absorb nutrients can be either dead or alive, and you see different kinds of terms used depending on if their food source is dead or alive. Fungi are dietary specialists, just like virtually every organism that you're going to learn about that's a heterotroph. And so, for example, organisms that specialize on dead organisms are just usually completely different from those that specialize on living ones, and even, what kind of dead organism. But you'll see various terms in your textbook that describe major categories. I think these are pretty straightforward and easy to wrap your mind around, so I will hold you responsible for these, even though I'm not holding you responsible for taxonomic names. I think these are actually far more informative and possibly even of greater interest to you. So what does this term mean? Saprophyte. Well, a saprophyte is a fungus that specializes on absorbing organic material from dead plant material. So these are the kind of fungi that would be then associated with plants that are dead. So for example, trees that fall over in the forest and are rotting, the saprophytic fungi then would be absorbing plant material from those dead plants. Now, because this is so different from the way that we obtain our nutrients, it's a little hard to think of how to explain this or make this more real to you. So if we just simply absorb nutrients as opposed to eating, then what would this be like? Well, imagine going out for dinner with a friend, and the soup comes to your table, and so instead of putting it in your mouth, you just kind of slap your hand in the bowl and then keep talking, because all the nutrients will just sort of be absorbed through your skin, right? You don't have to actually take it into your body. That's the life of a fungus, right? It's just totally through the body surface. But they still use digestive enzymes. So this point here, secreting enzymes, specifically we mean digestive enzymes. Because they're absorbing nutrients from outside the body, then they actually secrete digestive enzymes outside the body into their food, right? Totally different from us. We also have digestive enzymes. Where are digestive enzymes? Well, we secrete them into our digestive tract, our stomach and our intestines, right? So that when the food reaches those parts, the enzymes can break down those macromolecules into smaller, simpler nutrients that we can then absorb into our bodies. Well, this is sort of similar to the fungi, right? They're lying on some food, they're in physical contact with whatever it is they're going to potentially eat, but they have to then release the digestive enzymes outside their body. So to continue the restaurant metaphor where you're sitting there with your hand in your soup, this would be kind of like spitting saliva into your soup to get the digestive enzymes in there so that the nutrients would be broken up into a simpler chemical form so you could then absorb it through your body's surface. Of course, we don't actually eat like that, do we? So this is an extracellular digestion, and it occurs primarily outside the body, right next to the surface of the fungus, and then it can just suck the nutrients on in. Other kinds of terms that you will see throughout the chapter are, many of these are ecologically significant terms, but I think they're relatively user-friendly terms, and so hopefully you understand what they mean. Decomposers are fungi that are involved in decomposition, meaning breaking down organisms, right? And so fungi are an example of really important decomposers, in part because they have the enzymatic wherewithal to break down some materials that are very resistant to digestive enzymes made by other kinds of organisms, such as animals, for example. So fungi are capable of breaking down these remarkable structural compounds that are in the cell walls of plants, namely lignin and cellulose. This is pretty impressive. We can't do this, right? We can't just eat a chunk of wood and break it down digestively and get those nutrients out. We are incapable of doing this. In fact, it's only fungi and a few prokaryotes that are capable of doing this. Not all fungi, by the way, but ones that specialize in breaking down plant material have these particular enzymes. So the particular enzymes are listed here. The enzyme that can break down lignin is called lignin peroxidase. This is sometimes described as sort of a biochemical dynamite. It just takes this lignin, which is really tough to break apart, and just kind of blasts it into pieces. So it's not breaking it down so that the individual parts can be used as nutrients, but just to kind of get it out of the way to then enable access to the cellulose, which is a polysaccharide and a polymer, and so of sugar, a polymer of sugar, and so this can then be broken down by the fungus to result in organic molecules. And the enzyme that breaks this down is cellulase. Again, this should be familiar to you from Bio93. Anything that ends in ASE, for the most part, is an enzyme, and then the clue to what it breaks down is in the first part of the word, right? So cellulase breaks down cellulose. So fungi are really important in decomposition of plant material. Another way in which fungi make a living is as mutualists associated with all sorts of different kinds of organisms. What is a mutualist? Well, we'll talk more about various kinds of symbiotic associations. They are rearing their head in this chapter, so I'm going to go ahead and define these terms for you so you have a better feel for what this means, although you'll certainly see them again. The term mutualist specifically refers to a symbiotic association, and we've talked about symbiosis before, right? A very close association between different organisms. So this is specifically a symbiotic association that benefits both partners. So if you talk about a mutualistic association between a particular kind of fungi, specifically mycorrhizal fungi, which we'll talk about in a little bit, and plants, this means that they have a very close association with each other and they each benefit in some way. Okay, that's all I want to say about that. So, related to their role as important decomposers, fungi are a major player in the global carbon cycles. I don't know if you're familiar with what is meant by the term carbon cycle or other kinds of elemental cycles. What this refers to is the sort of chemical accounting that one can do if particular elements such as carbon appear in different different chemical form, and it is of interest to understand where is most of the carbon and what form is it, how is it converted from one form to another. And so this is somewhat similar to your figure 32-3 for your textbook. It makes exactly the same points. I just think it's a little easier to see what's going on. So CO2, of course, in our atmosphere is one chemical form in which we see carbon, right? And carbon is synthesized into organic molecules by what? Trees. These actually look a bit like gymnosperms. You know, just kind of looking at this, those look like needles to me. Anyway, those would be gymnosperm trees if they in fact were needles. But so at this point, then carbon dioxide is turned into organic molecules by the trees, including cellulose, which has then got the carbon locked up and inaccessible for most organisms on the planet except what? Fungi, right? So that's what down here it says glucose from the degradation of cellulose is transferred to fungivores, you know, such as insect larvae, ants, and squirrels, right? What does that mean? That the fungi can take the plant material, can break it down, can use the sugars, so now these are organic molecules that are accessible to the fungus, but anything that eats the fungus then also has access to what is now carbon in an organic form that is accessible. Ultimately, of course, when the glucose is burned up by the organism, then what happens? This releases the carbon again back into CO2. So that's the cycle. So they're very significant in terms of returning some of the carbon from being locked up in plant material then back into atmospheric carbon. Okay, I thought it would be nice to show you a movie rather than just looking at this as a static picture. I'll make it a teeny bit darker so you can see this a little better. So if you're in a forest then, so think about decomposition. These, of course, are green plants, so just ignore them. This is time-lapse because fungi grow fairly slowly. So we're looking at the forest floor here, and we're seeing various different kinds of fungi appear. And so what's the source of the carbon molecules that are used to make the bodies of these appearing fungal parts? Well, of course, these are the organic molecules that they are getting from it looks like these are all rotting trees, so they're pulling out the carbon organic molecules here and using them to grow. So anyway, I thought that would help you think about decomposition in a forest. But in addition to breaking down dead plants, fungi also have a lot of associations with living plants, and one of the very significant ones from the point of view of humans and plants are the so-called mycorrhizal fungi. This is the term right here. These fungi are particularly notable because they form very close association with plant roots. They are beneficial associates of the plants, and one can see this by comparing the growth of plants with and without mycorrhizal fungi associated with their roots. So this is figure 32-2 from your textbook, showing a plant that has grown with mycorrhizal fungi associated with the roots over there on the left, compared to an otherwise identical plant, which is much smaller than which was grown without mycorrhizal fungi. Notice that the plant on the right is still alive. It's not dead. It's not completely dependent on the mycorrhizal fungi, but it just simply grows more slowly. It just does not have access to the resources that the plant on the left does because of its association. You can't actually see the mycorrhizal fungi in this particular part of the figure, but if you look over here on the right, then here you can see a plant in the dirt in a sort of cutaway view. It's a little hard to tell where does the root system stop and the mycorrhizal fungi start, but because the root system is also a highly branching network, but so are the mycorrhizal fungi. And so much of this branching network that you can actually see in the soil for this particular plant are the associated mycorrhizal fungi associated with this plant. This shows a diagram that makes it maybe a bit clearer. There are two major types of mycorrhizal fungi. You are absolutely not responsible for knowing the differences between these. I think for this class, honestly, that it's important for you to understand what mycorrhizal fungi are, but not memorize the different terms because the similarities are much more significant between these different kinds of mycorrhizal fungi than the differences. One of the interesting things between these two different kinds of mycorrhizal fungi is they're completely different lineages of fungi. So this has this association and has evolved more than once in two different groups. But anyway, in both cases of mycorrhizal fungi, the fungi are very closely associated with the roots to the extent that they actually penetrate the roots. So this object here in the middle is supposed to be the tiny microscopic end of a little tiny part of the terminus, the very end tip of part of a root. And so you can see that in both cases, the fungi then extend into the actual surface of the root. The details of exactly how they do this are slightly different, but in both cases, there's a very close association. And in this figure, what is a little misleading is it looks as if the fungal network does not extend much beyond the root. You should not think of this ending right here, but rather it looks much more like this and extends for quite some distance in either direction. So we have this very close association with the roots. And we know that plants that are associated with mycorrhizal fungi tend to be bigger and grow faster, but what exactly are the plants getting from the fungi? In order to evaluate this, different kinds of experiments are needed simply than looking at growth rate and size of plant. There's some nice and fairly simple experiments described in figure 3210, so I'll take from your textbook, so I'll take a little time to walk through that right now. We'll start over here on the left. And so what these are experiments that are evaluating who gets what out of this association. So if we consider the simple design first shown on the left, here's the experimental treatment in order to tell is the plant passing on organic compounds to the fungi, that is, what are the fungi getting out of this association? You can do something like put labeled carbon dioxide in the air around the plant. What does labeled carbon dioxide mean? That means you take a less common isotope, so for example, if you're going to use a radioactive isotope of carbon, you would use C14, a much rarer form of carbon than the normal non-radioactive C12. So if you take carbon dioxide that has C14 as the carbon, then it is radioactive, and you can detect it with radioactive detectors. If you then have the plant surrounded by radioactive carbon dioxide, then if the plant takes some of this radioactive carbon dioxide out of the air and makes organic molecules with it, then the plant now has radioactive organic compounds in its body. Okay? And you know that the plant made these because you can follow the radioactive label. So if you do this for a plant with and without mycorrhizal fungi associated with the roots, so on the left here we have mycorrhizal fungi associated with the roots. Over here on the right we have roots without mycorrhizal fungi. And you can wait for a little while and then look to see where has the radioactivity spread. And so here's the results in a different panel of the same figure. Over here on the left, we can take the plant and take the fungus and look at the levels of radioactivity. And lo and behold, we have about 20% of the radioactivity in the fungus specifically. Obviously you have to separate them out before you evaluate this. And about 80% of the radioactivity is still in the plant. So why is there any radioactivity in the plant at all? Well, because it used the carbon dioxide to make organic molecules in the first place, right? And it looks like it passed about 20% of it to the fungus. Or did it? There's another possible explanation, right? Is that the plant takes the radioactive carbon dioxide and makes a bunch of organic compounds, and some of them just kind of leach out into the soil through the roots. in order to evaluate that, that's why the control on the right-hand side is so incredibly important, because if one then looks at how much radioactivity is in the soil, virtually none is in there at all. So clearly, it is not simply the case that the plant just kind of released or lost some of its compounds through its root surface into the soil, and the fungi just sort of grabbed them out of the soil. That is not the case. Clearly, it was passed directly from plant to fungus. So the clear result from this part then is that plants are actually providing fungi with carbohydrates, because you can follow the labeled compounds. Well, what about in the other direction? What are the fungi passing to the plants? If anything, you can do a similar kind of experiment, but now we're no longer expecting that carbon-containing compounds would be transferred to the plants, because after all, it's the fungus that's the heterotroph, not the plants. And so now labeled compounds are still being used, but now they're mineral, inorganic nutrients instead of organic compounds. So now the chemical element that's being labeled radioactively is either phosphorus or nitrogen. So now the experimental design is to add radioactively labeled phosphorus or nitrogen containing mineral nutrients to the soil in both cases. And similarly, where we have a plant with or without mycorrhizal fungi, with fungi on the left and without on the right. You wait a little while, and then you look and see where is the radioactivity. And after an appropriate amount of time, then here are the results. And one can see that, yes, in fact, large amounts of labeled phosphorus and nitrogen-containing compounds are in fact found in the host plant when you separate it from the fungi. Now, maybe it's just pulling them directly out of the soil and the fungus is not involved. This is where the control on the right-hand side is so important, because in fact, very little was found in the plant in the absence of the mycorrhizal fungi, which means that the plant then, in the absence of the fungi, was not able to take up as much of these radioactively labeled phosphorus or nitrogen nutrients. So very, very different. So then the conclusion from experiments like this is that, in fact, fungi provide plants with nutrients, but these are not organic nutrients. These are so-called mineral or inorganic nutrients, right? Nitrogen, phosphorus containing inorganic nutrients, not carbon. Mycorrhizal fungi, by the way, do not fix nitrogen. I often get questions about that. No, that's just the prokaryotes that do that. So those are all wonderful, beneficial things that fungi do, right? They make plants grow faster and more healthy. But fungi also have negative economic impacts. And so here is an example. This is figure 1 from your textbook, showing a couple of examples of how fungi can be associated with food crops, making them then unpalatable for humans. Notice the adjectives that are used here. These are significant. Over here on the left, it says parasitic fungi. Over here on the right, it says saprophytic. They're both on plants. What's the deal there? Well, what parasitic means is that it harms, or even kills some living organism. And so here on the left, we've got a corn plant, and here we have the ear of corn, and it's covered with this fungus. So these gray blobs here are in fact the fungus that is spreading over the surface of this, and so it is considered a parasite because this is a living rooted plant that is infested with this particular fungal type. Over here on the right, so saprophytic means, as I mentioned earlier, absorbing nutrients from dead plants. And so you might be thinking, well, why are these dead? If harvested fruits or vegetables like this The cells may be still alive, but they've been cut off the plant, and so these are considered somewhat different than if it was attacking oranges on, say, an orange tree that was still in the ground. So there is a distinction made. That's why it says saprophytic, because these have been harvested already. So fungi that cause rotten, dead, or harvested plant material then are considered saprophytic. And of course, we've all seen moldy food like this. It's really quite common. And what you may not have seen is time-lapse movies of mold forming. So let's go ahead and check this out. Here is an example of what happens if you have a bagel and leave it on a plate and just sort of leave it out in your kitchen. We've all sort of seen this. Obviously, this time lapse doesn't happen quite this rapidly, thank goodness. Notice that the actual species of fungus is identified right there. This is a very common genus of bread molds. And so there we go. Let's check something else out while you still have an appetite. So here is a strawberry, right? Leave a strawberry lying out in your kitchen, and eventually what's going to happen? It dries out a little bit, and then you see spots appear, and this is just kind of the beginning of the end, isn't it? Okay, it's going to start getting really fuzzy, right? Sheesh. Now notice this person does not know how to spell strawberry, but anyway. But they know how to spell the fungal species, okay? Got to give them some points for that. Okay, this is the fungal species that is infesting the strawberry. Notice this is different than what was infesting the bagel, okay? As mentioned before, fungi do in fact specialize on different food types, just as many other consumers of food products also specialize. We'll see this in the animals as well. We've also seen this in the protists. So if you think about what the significance of this is, or hopefully you're sitting there thinking, well, where did this fungus come from? I mean, we've been learning that fungus are not motile, right? So where do these things coming from? Well, in these particular examples, if something suddenly appears like this, then you can guess that spores, fungal spores, have landed on this strawberry or on the bagel in the previous movie and have then started to develop a fungus. So these are fungal spores that started this. So given that in your kitchen you can put out virtually anything, at least I'm speaking from personal experience, and it can grow mold and very different kinds of molds, then that tells you something about the fungal spore level in the air, right? So obviously this is coming directly out of the air, so about that. But anyway, and of course the other negative economic impacts of fungus, as we were discussing angiosperms, we were discussing the banana crisis, and this is in fact a fungal disease. It is a different strain than the one that wiped out a completely different and apparently even more delicious and fantastic banana that now none of us can enjoy because it doesn't exist anymore. That was wiped out in the 60s by a very similar kind of fungal disease that's now attacking the familiar Cavendish cultivars. So that's another fungal disease attacking those guys. But fungi don't just attack plants, they also attack animals. And so here's an example that's been on the news a lot. This is a new, relatively new syndrome that has appeared in bats in this country. That is the mammal bats, the vertebrate. And so here's a picture of a bunch of bats hanging in a cave. The syndrome is called white nose syndrome. This is a fungal disease. Over 1 million bats have died of this in just the last couple of years, primarily in the Northeast. There are many caves that are now closed to visitors because they're very worried about the spread of this fungal disease. It is obviously fatal. It is killing bats. And one of the symptoms is that you can see the fungus growing in the nose area of these bats. That's what these little red arrows are pointing to, is the fungal growth on the faces of these bats. But it's not just bats. You have also seen in the news a lot of stories about amphibians. And so this has been going on for decades. And so if you check out Wikipedia, for example, you can read about the acceleration of amphibian deaths in the 1990s that were first reported in the 80s are continuing to this day. There are a couple of different causes, but one of them is a really serious fungal disease, and so here it is described as a chytrid, a non-hyphal zoosporic fungus. This might make more sense in just a little bit, but this is something that has been linked to a lot of declines or extinctions of amphibian species in North America, Central America, South America, Australia, the Caribbean, et cetera, et cetera. So, 30% or so of amphibian species in the world is attributed to declines because of causes like this. So it's rather startling, and so you get news headlines like this, deadly frog bat plagues, eerie similarities. Actually, the fungus that causes white nose disease in the bats is a completely different fungal lineage than the one that is killing off the amphibians. But anyway, certainly they're in the news and they're a little worrisome. They don't all affect vertebrates. Here is a movie showing a particular ant that has been infested by a fungus. This is actually time-lapse photography. The ant is not moving because it's had this fungus growing inside it to the point where it can no longer move. You can see this growth of, this is an actual fungal part of the fungus itself, so this may remind you of what we saw in the decomposition in the forest floor, except this maybe seems a little bit more disgusting or scary because it's happening in an animal instead of on a dead log. But anyway, here is an extension. This is the fungus that is growing out of this ant. Here's another insect. Here are the fungal growths. Here's another one. These are static pictures. There's the fungus growing out. Here's another one that looks like little mushrooms, right? Here's another one. Look at this one. Sheesh, right? So this insect was killed by a fungus that sent up these little mushroom thingies, right? So, quite grotesque, huh? Some of these fungi, by the way, that are expert insect killers are in fact being explored as for potential use in biological control of pest insects. So I think that's interesting for you to know about. Let's go ahead now and consider the parts of a fungus, the fungal body form. And this is actually really straightforward. They have really simple body forms, which makes it very easy to learn about the basic anatomy of fungi. So this is not figure 32 from your textbook, but it's very similar. And so many fungi only occur in the very simple form shown here on the left, just single cells. Other fungi have just part of their overall life cycle as in such form. But anyway, when it is this very simple form, it's called yeasts. This is the same word that's used if you're a baker and you make bread and you buy baker's yeast. Yes, that is in fact a particular kind of single-celled fungus. Other fungi have bodies that look more like the picture on the right. And so this is what we saw, for example, when we were looking at the basic structure of mycorrhizal fungi associated with roots in the soil. We saw a body form that looked kind of like this, a mesh network, and that's the other kind of form. And so the multicellular forms of fungi basically have two different parts. There's the part above ground, which is usually associated with reproduction and usually only appears at particular points in the life cycle, and otherwise the rest of the fungus is completely underground, right? So here's the line of above ground versus below ground. Whereas below ground, there is a mesh network which looks quite different. But whether you're looking at the above ground part or the below ground part in a multicellular fungus, they're composed of a bunch of different filaments, which are called hyphae, that is plural. And so they're just arranged in a somewhat more compact form in the reproductive structures that form above ground, such as shown here, or above an animal's body for something that is growing in, say, an animal's body instead of soil. But the extent of the mycelium underground can be very extensive, right? Thinking back to the example of the fungus that's 2 miles across, that refers to a very extensive mycelium underground that is thought to be a single continuous individual that's very, very, very old. So that's how a fungus can be so large in extent, is that it can spread. Okay, because they can't move usually, virtually all fungi, but they can in fact grow. So that's how they end up in a new location. Okay. If we look at the structure of the hyphae a bit more closely, then they look like this. This is figure 32.5. These cells are sometimes referred to as compartments, either way. So this then would be a cell or a compartment. Why is a different kind of word used? It's because the separation between these cells and compartments is a little unusual. They often have incomplete or perforated divisions between the compartments or the cells, such as shown here. So these are considered then, pores, meaning that the cytoplasm between these adjacent compartments are continuous, at least through the pore. So they're all interconnected here and is pretty much continuous. So notice, by the way, that this kind of structure, this kind of filamentous network leads to a very large surface area when you consider that the significant issue for an organism that makes its living out of absorbing through its body surface, then the more body surface you can have exposed to the outside, the better from the point of view of absorbing. So having a body form that looks like this mesh network over here on the left is an ideal form for having a very, very large surface area, which greatly facilitates absorption from the environment. So let's now consider reproduction in fungi. And from the point of view of animals, or even plants for that matter, one of the things that's very unusual about fungal life cycles is that there's often a stage called the heterokaryotic stage. This is very unusual in that the cells or compartments within the part of the fungus that's heterokaryotic contains non-fused haploid nuclei from different individuals. So how on earth does this come about? Let's go ahead and check out the life cycle to make more sense of this. So this is a generalized fungal life cycle, which is figure 32.12 from your textbook, and shows the, by now, familiar characterization of life cycles in general, but this one is for fungi specifically. We've got asexual reproduction on the left and sexual reproduction on the right. So we'll talk about asexual reproduction first, because this is the easiest to understand. Notice in this particular case, we certainly have multicellular organisms, so this is not a life cycle plan that would be appropriate for a fungus that only occurs in yeast form, single-celled form here, okay? Because just looking at this, you can see multicellular entities in this. Okay, so let's start over here with asexual. Here we have a little bit of a mycelium here, or hyphae, right? And so it grows just by ordinary cell division. It gets bigger, and part of the structure can produce spores. These spores are virtually always haploid, as shown here. The spores can be dispersed in the wind. So this is dispersed. Right, so spores can just travel through the air, so ending up on your strawberries or your bagels or whatever. And then once they land, any individual spore can then undergo cell division and grow larger using the nutrients from whatever it has landed on, if that's an appropriate food source. Okay? And then once it gets bigger, it can form more spores, and that can go off and infect all the other strawberries in your pile of strawberries. In addition to the asexual reproduction shown over here on the left, there's also sexual reproduction shown here on the right. And I want to point out a couple of very unusual things that occur in fungi. Let's start over here with plasmogamy. You've probably never seen that term before. This refers to the fusion of the cytoplasm from different individuals. How do these different individuals get together? They don't move, right? Well, they would have to grow into each other, right? So they grow and grow, eventually bump into each other. If they're the same species but different mating types, so here we have one individual, here's another individual. Clearly they bumped into each other at this point here. Where they bump into each other, you can have fusions such that subsequent cells that are made, such as this one here, I don't know if you could see it, I'll make it bigger over here. Subsequent cells, post-fusion here then, have haploid nuclei from the two different individuals, because we're talking about hyphae that were originally haploid. So they had each cell or compartment had its own little haploid nucleus. We have plasmogamy. We have this heterokaryotic mycelium now, where we have two different haploid nuclei. This one's N, this one's N. They're in separate nuclei still, so that's why this is referred to as N plus N, which is completely different from being diploid, having a single nucleus with two copies.